So can you see the shared screen? Is it visible?
Yes, sir.
Okay. So this part we have started in the previous class, and before I move to my this class, I would like to know whether you have received all the PowerPoint which has been uploaded. I have uploaded all these slides of up to carbohydrates. Have you seen any of these slides? It is basically a PowerPoint which has been uploaded. Anyway, if you have not checked it, you can check it. All the lectures till date has been uploaded. Now coming to the previous class where we have talked about what are the proteins and what is their structure and function. This is what I have started. Now looking at the function of the protein, you have learned that proteins, they are most versatile macromolecule and they have certain crucial functions. And these crucial functions we have discussed. Now we are moving on to what are the properties of proteins. So proteins are linear polymers and they are built of monomeric units called amino acid. So if I compare from carbohydrate, you have learned in the carbohydrate, the polysaccharides, they are made up of monomeric unit, which are saccharides or sugar residues, and they are having a specific kind of a bonds, which we refer as a glucosidic bonds or glycosidic bonds. On the other hand, proteins, they are made up of linear polymer of monomeric units of amino acids. And these amino acids, they are joined together by a specific bond, which is called as a peptide bonds. Now, these proteins contain large number of functional groups. Mostly in the carbohydrate, you have seen a very common functional group is a hydroxyl group. Here, in case of a protein, you will find that hydroxyl group could be functional group, imide group can be functional group, carboxylic group could be a functional group. So you will find different types of specific groups which are functional group present in the protein, and that is because they are made up of amino acids, and amino acids, they have these functional groups. The proteins, they can interact with one another and with other biological macromolecules to form complex assemblies. One of the examples you have learned in case of a carbohydrate, where protein can interact with the carbohydrate. The kind of interaction you have seen is that protein can bind with either carbohydrate, they can have non-covalent interaction with the carbohydrate. On the other hand, protein can also have a complex with the carbohydrate where you have a covalent bonding. So don't get confused. Whenever you have a glycoprotein or proteoglycan structure, they are having a covalent bond. On the other hand, when you have lectins interacting with the carbohydrate moieties, the interaction could be non-covalent interaction. So these are two different examples which shows the protein can interact with different other molecules. I'll be talking about the interaction of the protein with the DNA or nucleic acids. You will also learn the proteins can also interact with the lipids, etc. So these are different interactions, and that's where the protein plays a major role in their in cell function. Some proteins, they are quite rigid, whereas others, they display limited flexibility. Now, when I say rigid, what does it mean? Rigid means their structure is a more, fixed one. They are more of a structural molecule, whereas some other proteins, they have limited flexibility. That means they can change their structure. This is a very interesting point that proteins, they can change their structure after binding with certain ligands, after binding with certain other molecules. And this structural change or this flexibility in the molecule is a functionality. Now, down on the lowest part of the slide, I have shown different types of structure of the protein. First one refers to the primary structure. Second one is a secondary structure. Third one is a tertiary structure. And so I'll just try to emphasize it. Yeah. So this structure which you see is the primary structure of the protein. So in the primary structure is a definition where amino acids are linked together, and you can see that there are different circular structure. These are each individual amino acid. When they link together, they form, they link with each other, form peptide bonds. So this structure is known as a primary structure. Now, the complexity starts from primary structure to secondary structure. All these details we are going to discuss in in future slides. But let us first talk about what is the basic concept. So you have a protein which are linked together by amino acid. So monomeric units are amino acid. Then these amino acids, they start interacting with each other with the neighboring group, and then you get a helical structure, and this helical structure is referred to secondary structure. Now, once these helical structure, they are interacting locally or with the neighborhood, it is a possibility that some of the groups which are far away, they come closer to each other, and that leads to the structure which is termed as a tertiary structure. Now, tertiary structure on its own is totally functional. What can happen is that different tertiary structure, for example, this is the one, when they start interacting with each other, they form a quaternary structure. These details we are going to discuss further, but it gives you a very basic idea that the proteins, they can have different level of structure. So it is a kind of hierarchy which we will discuss. Now, before we move on to their structure, first most important thing is to understand what are the building blocks and how they are made up of. So these building blocks are amino acids. So when I say amino acid, it is very clear that you can make out what is this. It says on one hand amino, on the other hand acid. So it has amino group, that is NH2, and it also have carboxylic group. And because of this, these amino acids, they have a unique role in the functioning, and we will talk about that. So when I take a amino acid, any amino acid can have a carbon. This is the example. You have a central carbon. On one hand, it is attached with a hydrogen. One side you have amino group. I have shown positive charge. I should describe why I have shown positive charge. One, you have carboxylic group. And so in all the amino acid, you will have a carboxylic group and one amino group. This is common. And this is also common that on one hand, you will be having hydrogen. So these three things are fixed. What is a variable here is the variability occurs in terms of the variability occurs basically in terms of the R group or the group which is attached. So the CH3 group, which is attached to the amino acid, this is the one which makes a difference between different. So what you have is you have carboxylic group, amino group, and that's why we call this amino acid. So this is refers to amino, this refers. And this whole chain, this part, is known as main chain when they are interlinked together, and this part CH3 group refers to the side chain. I will discuss this when we talk about the protein structure. So let us go back and see. So these amino acids, they have a central carbon atom linked to an amino acid, a carboxylic acid group, a hydrogen atom, and distinctive R group. So R group is different. So if there are 20 different amino acids, all of them, they will have first three common, and the difference will occur because of the R group. And the R group, as I said, is referred as a side chain, whereas the rest part is referred as a main chain of the amino acid. So this This is the main chain when they are forming a protein structure. Now, as we have discussed in the carbohydrate, you recall that carbohydrate has got stereoisomerism. So they have different isomers. They are L isomers, they are D isomers. And one thing which was very common in carbohydrate, that all naturally occurring carbohydrates, they have a predominance of D form. So whenever we have talked about glucose, we said D glucose. Whenever we talked about galactose, it is a D galactose. So what does it mean? When I'm saying capital D, it does refer to the orientation of hydroxyl group in case of carbohydrate. Same way, you have the based on the orientation, these amino acids, they are also referred as either L isomer or D isomer. Interestingly, you will find while carbohydrates, they have all the naturally occurring carbohydrate, they are the stereoisomer. Whereas in the protein, all the amino acids present, they have L stereoisomers. So there is a difference, but this is a natural selection process by which they have been selected, and that is what it is. Though there are some D isomers of amino acid, they are present, but they are rare. You may have learned them in the bacterial wall, but I'm not talking about the exceptional one. I'm talking about the amino acids which are present in the protein structure, where they are, contributing the structure of proteins. Now, coming to the amino acid, I have also written alpha amino acid. So just I will go back to the previous slide. Okay, not this one. Yeah, this slide, next slide. What you have is that this is the carbon atom. You have alpha carboxylic group. This is your uh, the carboxylic group is alpha carboxylic group, whereas the amino group is also alpha amino group. So this is carbon atom. This is alpha carbon atom. Here amino group is attached, so we call it the alpha amino. And here carboxylic group is attached. So to the alpha carbon atom, both the carboxylic group as well as amino groups are attached, and that's why we referred them to alpha amino acid. Now coming to the D form or L form, you can see that the orientation around the carbon atom, which is given over here. Oh, this, so if I look at this amino acid, what you see is that there is a specific orientation of different groups around this carbon atom. So this is alpha carbon. Here on one hand, H is attached, which is a fixed one. R is attached, which is a fixed one. Amino group is attached, which is a fixed one. Carboxylic group is attached, which is a fixed one. So all of them, they are fixed one. Now what I'm looking at is when the amino group is present on the right hand side, we call them as a D isomer. And whenever it is present on carboxylic group is present on the right hand side, we call them as a L isomer. And what you find is that in protein, all the amino acid which is present is in the form of L isomer. So looking at this, you have learned few things. One is that the amino acids, they have a common architecture in terms of their bonding, in terms of their groups, except that this R group which we are referring to, that is going to be different. Now you will find that the L isomer, which is the most common, that is the one which predominates. Other than that, another interesting finding you will have is that these amino acids, because they have amino group on one hand and carboxylic group on the other hand, that is why they will be dependent on pH. So what will happen? At neutral pH, they will exist as a zwitterion. That means they are dipolar ion if the pH is neutral. So that is why you find that I have shown you the amino group positive charge and carboxyl group negative charge. So under neutral pH, both the groups they are charged one. But what will happen when there is a H ion, when there is a more proton, that means at lower pH, this CO negative will get neutralized, and because of that, you will have the amino group which will have a positive charge. So the protein will have a positive charge at lower pH, and what will happen at higher pH? So when I'm saying protein, I'm also referring to amino acid. So if amino acid is a positively charged, that will give total charge on the protein as a positive from that particular contribution of the amino acid. Now, same way, if I go to high pH, you will have more OH ion. These OH ion will remove the proton from amino group and they will leave them to have a carboxylic group. So at higher pH, amino acid will have negative charge. So this deuterium ionic structure of amino acid or the charges in the amino acid gives them flexibility to change their charge on the surface, and later on you will learn that this change in the charge on the amino acid, which could be individual amino acid, leads to the change in the overall structure of the proteins. So when we are talking about this building blocks of the amino acid, which are building blocks of proteins, which are amino acids, what you are learning is that the behavior is unique behavior. Now coming to the different types of amino acid, you will have 20 types of amino acid. Now when I'm saying 20 types of amino acid, I'm referring that H will be fixed, amino group will be fixed, carboxylic group will be fixed. There are 20 types of R groups. So let us focus on R group. So how they are variable? They could be variable in their size, their shape, their charge, their hydrogen bonding ability, their hydrophobic characteristics, their chemical reactivity. All of them, they may vary. So we will talk about amino acid. We will take them in a groups where they have some similarity, and then we will discuss about their property. So here we are going to talk about a set of amino acid where R group. So let us see what are the R group. This is the R group. The R group here is what you can infer from here. What is the R group here? This is R group. This is R group. So you see there is a difference between glycine and alanine where H or hydrogen atom is replaced by CH3 group. Then in case of valine, the R group is slightly bigger and it is a branched also. Then leucine, it is further, different. But there is a commonality also. So what is a common over here? Let us talk about all this which is a common. What you see is that it starts with a simple hydrocarbon side chain. Now this hydrocarbon side chain could be variable in their size because it starts with the smallest one, that is a proton, and then moves on to slightly bigger one where you have a methyl group. Then you have a branching and you have, valine. So this is a branch chain. Then you have a leucine where again branching is there, but the number of carbon is more. Then you have another isomer of leucine where the branching is slightly different, that your methyl group is slightly away from the is slightly closer to the main chain where it is slightly away from the main chain. So we will talk about that. Then you have a methionine which has a sulfur group. I will talk about that differently. But it is again at the terminal you have a methyl group. On the other hand, there is an amino acid which is a proline. This proline has, if I look at the structure, look at the structure, you have a carbon here, amino group here, proton here, like any other amino acid, and then the side chain, which is 1, 2, 3 carboxylic group, has come and merged with the interactive with the amino group. So this side chain has joined the amino group which was present, and that is where this amino acid is unique, though it has been under the amino acid. But can you tell what is this? This is actually not amino acid, this is amino acid, because you see here one of the proton is less compared to these amino groups. So here you have NH2 plus compared to any other amino acid where you have NH3 plus. Now let us look at individual amino acid and discuss a little bit about the chemistry. Look at the glycine. In glycine, what you have is will it be stereoisomer? Yes or no?
No, sir.
Okay, because this is the only amino acid where you will lack the stereoisomerism, because its R group is having hydrogen, which is so the around the carbon atom you have a two groups similar, and that's why this is the one which is not a stereoisomer. Other than glycine, all of them they will show stereochemistry because carbon is joined with all four different groups. Now, alanine, what kind of a group it is? Can it form hydrogen bond? Can it have positive charge? Can it have negative charge? So here, these groups which I have shown to you, all are hydrophobic in nature. So all of them, they have a hydrophobicity. So that's a very common that all the groups which are present, they are having hydrocarbon side chain. So you have glycine, alanine, valine, leucine, isoleucine, methionine, and proline. All of them, they have hydrocarbon as a side chain. So suppose these amino acid is present in a protein, what kind of interaction you will have? You will have hydrophobic interaction. Most of the time, they will not be present on the surface because they cannot interact with the water. And if they are present, they will be squeezed on the inner core of the protein. So it's a very interesting phenomenon. We will talk a little more, but at this point, it is more than enough for you to understand that these amino acids are having hydrophobic side chains. Now coming to the another group of amino acid which will have aromatic side chain. So let us start with the simplest one, the phenylalanine. Uh, okay, this phenylalanine structure is the one. Let us look at this structure. Up to here, everything is common. Now, if I put this CH2 group or CH3 group, it becomes alanine structure. And when I add phenyl group, we call it the phenylalanine. So it's a very simple structure. If you remember the basic core structure of amino acid, where carboxylic amino group is present and CH3 group leads to the alanine structure, adding the phenyl group gives you phenylalanine. So it is a diphenylalanine. Now, every time, even in the previous slide, I have given you a code. So each amino acid is referred by three-letter code, which is a glycine, and single letter code, which is given by the single amino acid. The reason why single amino acid code is important or why three amino acid code is important is that when I give three amino acid code, even if there are two amino acids starting with the same letter, you will not get confused with them. So when we give the single letter code, you will slowly learn that if the amino acid is repeated, for example, glycine, and you will see later on glutamic acid, their single letter code has been changed. It is not necessary that it should start with their initial name. So this is a very simple thing. Now, initially, most of the amino acids, they were named with the three-letter code, but when people, they started looking at, making the sequence of amino acid, it became very cumbersome. It was 3 times more than the expected numbers, and that is why single letter code has come. So if I want to code something, or if I want to write a code of the amino acid, it becomes very difficult with three-letter code. But still, in some books, you will find three-letter code. So I have given both three-letter code and single letter code, and that is what you will find in most of the books. So I have talked about phenylalanine. Now looking at the tyrosine, it has got one additional OH group than your known phenylalanine structure. So this structure, up to here, everything is same. this OH group is extra present in tyrosine compared to phenylalanine. While tryptophan has a very different structure, so up to here you have up to here what you have is you have a structure of alanine and then a group which is attached with this gives it as a tryptophan structure. Now all the amino acid has got aromatic side chain. Because of the aromatic side chain, these amino acid they will show hydrophobic characteristic. They will have a pi-pi interaction. What you find is that if I compare phenylalanine with the tyrosine, tyrosine will have lesser hydrophobic structure because of presence of this hydroxyl group. Then we have another set of amino acid which has a hydroxyl group in the chain, and if you recall we have alanine structure. So alanine has got similar structure like serine, except that serine terminal is OH. So if I compare alanine and serine, serine will become more hydrophilic because of presence of hydroxyl group. Same way threonine, it is a branch chain structure, it is a long chain structure compared to the your alanine or serine, and here what you find is that one additional carbon is there and one hydroxyl group. So whenever there is a hydroxyl group, they will have a tendency to interact with other and their reactivity increases in terms of interaction. One very interesting thing you can recall from the previous lectures that whenever the interaction of amino acid with the carbohydrate occurs and you have O-glycosidic linkage, these are most common. Serine is one of the common amino acid, or threonine could be a common amino acid, which allows the interaction between between the amino acid of a protein with the carbohydrate. Now coming to another type of amino acid which has got SH group, sulfhydryl group. This is known as a cysteine. Now cysteine is the one which has same structure. Up to here the structure is same. If I look at the structure up to here, it becomes alanine structure. Now in alanine what we have done is that we have added SH group. So adding this thiol group, it makes cysteine very reactive. It becomes much more reactive than the other amino acid. What could be the possibility of the presence of cysteine? If there are two cysteine residue present in the neighborhood, they can react with each other, and what they can do is that their SH group can interact. If two SH group they interact with each other, they form SS bond. So you will have two cysteine residues interacting with each other, they will form disulfide linkage. It's a very strong, it's a covalent linkage which is present in the protein, and that is coming because of the interaction of the cysteine. These are set of amino acid which has got basic. So here, interestingly, what you find is that each amino acid, I just go to each amino acid, they are having one amino group and one carboxylic group. But these amino acid which I am referring to, basic amino acid, they will have additional amino group or additional positively charged group which is present in these structures. So these are the amino acid which is lysine, arginine, histidine. So these three amino acids, they are a basic amino acid. One is lysine, arginine, and histidine. What you find is that here the chain is also relatively longer chain, and they will have one additional amino group or one additional positive charge on the amino acid. This histidine is the one which we will discuss later on, where you find that these amino acid they are having their pKa value. Do you understand what is a pKa? What is a pKa? You have learned about the pH.
So negative log of
Okay,
K.
negative log of. So what it refers is that it refers to the the amino acid or any molecule which is whether in the charged form or it is a protonated form. So that is what it shows. So when I say pKa value, that means at that particular pKa value, at that particular pH, they will have they will so if I look at different acid, they will have their pK value contributed from these amino group, this carboxylic group, this amino group. So they will all contribute towards the pK value of the amino acid. So you will be learning that the dissociation constant for this amino acid and this amino acid varies, and that's why the pK value contribution of each one will be different. Now, why I have talked about the histidine is that the pK value of its side chain is something like 6.5. What it refers is that at neutral pH, they have very close proximity to switch to either positive charge or negative charge. So they are in a in a domain where they can switch either to have a positive charge or the negative charge. And this is an advantage which you learn, that that is what is required in many protein when they have a multifunctionality. Then I have a set of two amino acids which are having carboxylic group, same structure. So here, up to CH2 group, it is alanine. One carboxylic group, it becomes like aspartic acid. When you have one additional CH2 group and carboxylic group, it becomes glutamic acid or glutamate. Now, what you have here is that these two amino acids, they will have a negative charge. So what you have one amino positive contribution, one negative contribution from this carboxylic group, and one additional carboxylic group. So whenever these basic amino acid is present on the in the protein, they will charge towards the positive charge. Their contribution will be for positive charge. On the other hand, when these amino acids are present, like acidic amino acid or the amino acid having negative charge, they will contribute towards the negative charge in the protein structure. Another very interesting thing is that these amino acids, like aspartate and glutamate, they can form amide bond reacting with NH3. You will have CONH2 at the terminal end. So these are not contributing toward the charge, but they are the derivatives. So aspartic acid can become into asparagine, and glutamic acid can form glutamine. So they become part of the amino acid series. So what we have talked about so far is that we have talked about 20 amino acids which are present in the protein, which is a naturally occurring macromolecule. Now, till this part, do you have any question? Any questions so far?
So I wanted to know why is it like that that all proteins are made up of only the L forms of the amino acids?
Okay, I will not be, this is a natural selection process. And if we look at that, all the carbohydrate, they have taken orientation of D form, whereas all the amino acid, they have taken orientation of L form. Now, question could be that when the selection has occurred, can we have L form of amino acid in the organism, which is highly evolved organism, and the D form of amino acid in the lower organism, or the organism on which we feed on? So you can say, for example, let us think of a scenario. And before we think about the scenario, the condition here is that when I talked about, I said all the amino acid, whether it is present in the proteins coming from plant or coming from animals or in our own system, all of them, they are made up of the amino acid, which is L form of amino acid. So whatever we have discussed so far, all the 20 amino acid, they are in the L form. They are not in the D form. I will also talk about that there is one amino acid which you find in case of microorganism, they are T, and we'll talk about the logic. But before that, let us say, and let us assume, take a hypothetical situation where the amino acid present in the plant are D form of amino acid, and the amino acid which is present in the higher organism like us, who are feeding plant protein, who are eating plant protein, have L form of amino acid. So what would be the problem? The the problem would be that D and L, they are two different stereoisomers. And when they are interacting with the enzyme or the catalyst present in our body system, if they are two different forms, they have to be interconverted. So what I mean to say here is that suppose I'm taking a protein from plant. So you are taking pulse, you are taking, another plant protein like wheat, you have gliadin and other proteins. So when I take this protein, when I eat it, that time, if all the amino acid, of course it is not, I'm just taking a hypothetical scenario where these amino acids are having D form. So suppose I have a D lysine, D arginine, D histidine, D aspartic acid, and that is making a protein. And now I eat it and I want to digest it. What will happen? We need a set of enzyme which should degrade a protein having D amino acid. Now, once we form the D amino acid in our body system, all the D amino acid, because they cannot become part of our body system, so they have to be first converted into the L form, because that's the way our body system has been, evolved. Now, if I convert D form into L form, you need a set of new enzymes. So suppose I have a D-alanine, which has formed from the plant protein degradation. Now D-alanine has to be converted to alanine to become part of our body protein. So that is why what you find is that nature has selected in such a way that all the amino acids, they exist in one form, that is L form. Same way the stereoisomerism, and we will talk about the enzymes, and in enzyme you will say that they are very specific. If they work on the D amino acid, they will not work on L amino acid, and vice versa. So what you require, if I have a protein which is the D form of the containing amino acid, which is the D form, and we have to synthesize a protein which has L form of the amino acid, the cost of energy, the synthesis of the protein, the synthesis of enzymes, that will require huge cost of energy. And because of that energy conservation, this is the nature where you have all the amino acid present in the plant, present in the microorganism, all of them, they are in L form. Is it clear to all of you?
Yes, sir.
Just give me
Excuse me,
some
sir.
clarity. Yeah,
So I had a doubt. In bacteria, we observe that
continue to
peptidoglycan.
that. Yeah,
Hello,
just tell
am
me.
I
Yeah,
audible?
yes, yeah,
Yes, sir.
I can hear
So,
you.
so in bacteria, we observe that the peptidoglycan consists of D-amino acids in the
Okay,
D-alanine and D-glutamic
right,
acid.
right,
So,
right.
sir, how are these layers
Huh.
degraded? If
Okay, that is where it is important. Now, what you want when a bacteria has evolved, bacteria evolution is slightly different than the evolution of other organisms. So initially, if you find most of the amino acid present in the bacteria, all the proteins present in the bacteria, all of them, they are made up of L amino acid. But some of the peptidoglycan layer, which has mostly your D-alanine, is present over there. Now, the logic why the D-alanine is present is because bacteria try to evade the enzymes which is present in our body system. Suppose bacterial cell wall or peptidoglycan layer is also made up of L amino acid. As soon as you eat or ingest bacteria, they will be treated like another protein and they will be broken down. So in order to protect itself, bacteria has evolved an integrated D amino acid so that it becomes your body enzyme becomes inactive. Now you may ask how it is proven and how it is, let me tell you that there are certain antibiotics. Do you understand what is antibiotic?
Yes, sir.
so,
Okay, you eat it. Sometimes your doctors, when you are infected, if you want to, if you got the bacterial infection, doctors give you antibiotic. Now, some of the antibiotic, you will see that they are peptide in nature or, made up of amino acid. And these amino acid will have D amino acid. So there are exceptions where D amino acid is present, and these exceptions are the bacterial wall and the your antibiotic. And the reason here is that the in case of antibiotic as well as in the case of bacteria, they want to avoid the enzymatic degradation when they are ingested. Is it clear? Hello?
Yes, sir.
Yes, sir.
Okay. Any doubt? No. Okay, so this is giving you some basic idea that how these organisms have evolved. So this is a very common question. Why all the amino acid present in all the organism or universe, they are having similar kind of stereoisomerism. And the logic is very simple, that we do not want high cost of energy. Why high cost of energy? Because if I want to process a protein which is present in the in a plant, and I want to process it for human being, which is a very common thing, like we eat pulse, we eat a lot of other proteins, which is a plant protein, or we eat bacterial protein. So whenever we are processing them, it should not become resistant. So our enzyme should process them, and that is why all the plant proteins, all the microbial protein, except that we have talked about, like bacterial cell wall, which is for the reason of protection, all of them, they are made up of L form. Now, let me tell you here, most of the time in my class, you get worried that there are so many structures, so many, things, how we remember them. Most important thing which you have to remember is a class. And this remembering class, that means all of them, they become. So if I say the basic amino acid, you must remember what is a lysine, what is the arginine. You should also have an idea that lysine will have a long chain. There is one thing which I have slightly missed in the previous slide. If I look at two structures, see, this is a leucine structure. I'm sorry. Let us take a pen. There are two structures. This is a leucine and this is isoleucine. If I look at their carbon atoms, hydrogen atoms, all of them, they are similar. There's more or less, you can count them, their number is same. The only difference, what difference you see here? The difference here is that this CH3 group is far away from the main chain. So if I say this is a main chain and this is a methyl group, it is away from the main chain. Whereas this methyl group is present close to the chain. So if I ask you that if I take a leucine and make a polypeptide, a polyleucine, and another case I take polyisoleucine, I take both of them, put them in a hydrophobic solvent. What will happen? What should happen? So it is a long chain and I'm putting them in a solvent. What they will do? They will start interacting with the main chain. Now what you find is that, or if so what will happen, this chain methyl group is far away from the main chain, and that is why they will not stop them from folding. Whereas this methyl group, which is very close to the main chain, it will provide steric hindrance when they try to fold. So these type of things, they happen. And one more interesting question comes, and that is what we should discuss here, is that you have in this class, I have discussed so many amino acid like valine, isoleucine, and all of them, they have a common feature. This common feature you see is that all of them, they are hydrophobic in nature. So why nature has made all of them having hydrophobic group? Why not in nature has selected only alanine or only glycine? Or if you say that there is a requirement, they may have selected, say, leucine. So why it is so? This part you think over, we will discuss about it. So this gives you a basic idea that how different amino acids are present in a protein structure. We will discuss more about it. And this is absorption spectra. We will talk about the absorption spectra. So I will give you 1 or 2 minutes because I have another class at 10 o'clock. So if you have any question, you can raise the question. So one thing which I am